So with that, I actually have the pleasure now of introducing my next colleague, Mohammad Albanna, and for him to talk to us about the promise of regenerative medicine.
Thank you very much, Scarlett. Problem. Very cool. Good morning, everyone. Thank you very much for having me. It's a great pleasure to be at ASU here. I moved from North Carolina almost 5 years ago, working in a hospital, and now I moved actually to the industry. So I'm working now on a new startup that I'm going to tell you about it at the end of the presentation. But something that I would like to share with you here is about the regenerative medicine. I've been working in the field of regenerative medicine for almost 15 years right now, trained under the guy who really pioneered the field, Dr. Anthony Atella, who created actually the first human organ in a lab back in the early 90s and put it back in a patient, where the whole field actually started. We probably heard a lot about the regenerative medicine over the last probably 5-10 years. We heard about stem cells. We heard about tissue engineering, and recently we are actually hearing about 3D bioprinting. And every day we probably hear or read an article that talks about curing diabetes in rats, or printing a liver in a lab, or actually like what happened back in April, the first 3D printed vascular heart. So where is that? Where is that is going to take us? We hear about stem cells, that they have the ability to cure this disease or improve the quality of patients. So the question that I always get is, this is a hope or a hype? So I might not be able to answer this question for you by the end of this talk, but I would leave you with a lot of things to think about and maybe make up your mind on how to answer this question. So I'm a CEO and founder of a company here in the Valley, started 1.5 years ago, called Humabiologics. I will tell you about it a little bit toward the end of the presentation. I'm also an adjunct assistant professor of general surgery at Wake Forest Baptist Medical Center. I completed a fellowship in burn surgery 10 years ago, and I run clinical trials around the US for new products that aims to contribute to wound healing or skin regeneration. So as I mentioned, we heard a lot about the regenerative medicine. We heard about the stem cells. We heard about the 3D bioprinting, the tissue engineering. So what it's really the regenerative medicine, or what's the goal of the regenerative medicine? The cool thing about the regenerative medicine that it is a multidisciplinary field, where engineers with different backgrounds, mechanical, structural, civil, mechanical, electrical, computer, they can work in this. Scientists, cell biologists, molecular biologists, you name it, lawyers, business people, quality assurance people, multidisciplinary field that actually brings all those expertise together for one goal, to either actually repair, replace, restore, or regenerate a missing function of a tissue or an organ. So any form of that collaboration can actually happen to basically achieve that goal. So what we are trying to do in regenerative medicine, if there is a missing tissue there or a missing function of an organ, what we are trying to do at that point is to put it back and basically restore that missing function. When it comes to structural tissues, the issue might be not as complicated as we always think. When we are dealing with, let's say, a second or a third degree burn injury, it happens to the skin, which is a structural tissue. It's not a life-threatening condition unless it's an extensive burn injury above 40-50% total body surface area. But if it's a missing structural tissue, the solution for this is basically through a skin grafting. And the early development of the skin grafting was actually happening through taking a skin from a deceased donor, remove the cells, and end up with that piece of tissue and graft the patient with it. And then actually that took off, so we started actually taking tissues from animals. We are taking pig skin, pig intestine, cow skin, any other tissue that we can actually get from the animals. We strip off the cells and we graft the patients with them all FDA approved. We even actually take skin from the fish, and it's FDA approved, and we actually graft patients with it. The early development, actually, this is not a new idea. It probably goes back to 100 years ago when they were actually using the amniotic membrane after a delivery to graft patients and wrap wounds with it. So that's kind of like, the development of the extracellular matrix and tissue augmentation. If we need to include cells, maybe this is also like, possible. Again, for structural tissue like skin and bone, that might not be as complicated as we would imagine. You can actually combine the cells and the bone material and do what we call it a tissue augmentation. So you're basically patching that skin or that bone. But really the issue gets more complicated when you are talking about functional tissues or functional organs. So the basic concept of tissue engineering or regenerative medicine is basically to take a cell from the patient, grow it in the lab, end up with billions of cells, and then combine it with a biomaterial or a scaffold and basically try to do a conditioning adaptation for the cells with the new biomaterial and put it back in the patient. So with some tissues or body parts like an aortic heart valve, you can actually use a mold to create that biomaterial and then you can put the cells on it. For complicated organs like liver, solid organs in general, kidney, pancreas, liver, the case might be actually not as easy when it comes to making that organ. So until this moment, actually, we are utilizing animal organs or human organs, and we remove the cells and we put the patient cells back on them. The only problem with that is when it comes, for example, to the kidney, there are at least 23 different type of cells in the kidney working together. So defining those cells and putting them back in the exact location is actually a complicated thing. Keeping that tissue vascularized and alive, that's actually also complicated. So that's where we actually started utilizing different technologies to fabricate those tissues and organs. So at the end of the day, we are actually introducing the regenerative medicine as combining cells, growth factors, and biomaterials together, and boom, you have an organ or a tissue. Actually, it's a little bit more complicated than this. As I just mentioned, identifying what are the cells and how to put them together and keep that tissue alive is basically a challenge that we did not address until this moment. However, there are a lot of technologies that actually contributed to the field of regenerative medicine, and one of them that I'm actually we worked on it more than actually 10 years ago. It's a technology that we used to treat wounded warriors in Fort Sam Houston in Texas. So we actually brought this technology from Australia, and we actually developed the clinical trials here in the US, and last year it actually got approved by the FDA. The technology is neat. It's a bedside kit that actually take a small biopsy from the patient's skin, mince them, and then actually spray them back on the wound. What's neat about this technology that actually from a small size biopsy, you can actually cover 80 times surface area. So you don't need a lot of skin for extensive burn injuries actually to treat that wound. You need a small biopsy to cover much bigger area. I apologize for the pictures. You might see some pictures in my presentation, but this is actually the result of such technology, actually, where you have a massive burn on a kid, and within actually a couple of hours, you are actually able to spray the cells, and you can get actually a near perfect skin regeneration that's actually going to last with that kid for the rest of his life. We also made a lot of different advancements on different tissues, like actually the first human blood vessel in a lab. This is actually going through late stages of clinical trials, and hopefully it's going to be approved within the next couple of years to give actually patients new blood vessel that actually made in the lab, rather actually than going and taking a blood vessel from another location and put it back. You are actually making a custom-made personalized blood vessel for those patients, creating them in the lab, then putting them actually in the patient. So that's actually when it comes to tissues and organs, like blood vessel and like, skin and bone. But when it comes to organs and mainly solid organs, again, things get complicated. As I mentioned earlier, the first actually human hacking was actually done by this gentleman. His name is Anthony Attella. He's the chief of urology at Wake Forest Baptist Medical Center, and he leads actually the largest lab in the world for regenerative medicine in North Carolina. So this guy actually created the first organ in a lab, which was actually a bladder. He actually got a biomaterial, made a ball out of it, went to the patient who was actually a baby at that point, took a biopsy, grow the cells in the lab, and put them back into that biomaterial, and then put it in the patient. The patient now is actually in his early 30s. He's actually a practicing lawyer in the state of New York. So that was actually the first successful story of creating a human organ in a lab. And since then, the whole field actually started expanding. So again, for tissues and organs, there are some of them, they are easy to make. For example, when you look at like, spherical or hollow tissues or organs, that's actually easy to make. But when it comes to solid organs or more complicated tissues, then we need actually a new way of fabricating those tissues. And here is where the 3D printing or 3D bioprinting actually comes to play, where we can actually print materials and cells in a layer by layer fashion to and put the cells in the right place and let them talk to each other the way that they have to. So again, the concept of 3D bioprinting is very easy. You deposit layer by layer, and whether you are using cells, materials, or combination of both, or any other platform. So this is actually some of my work back in 2006 when we actually were using an HP desktop inkjet printer to actually come up with the field of bioprinting. And that was actually the idea of Tony Attella when he actually brought us to his office, and he asked, can we really print tissues and organs? At that time, 2006, a lot of people, they didn't have access to regular printers to print documents. So at that point, we were actually thinking of, can we take this to the next level? So we literally took the ink cartridge, cleaned it up, put cells materials in it, and we started printing. I don't know, the video, if we can play the video here, it's actually very cool to see how it all started. So you'll see here, can you start the first one, please, this one. So this is actually when we were printing a heart. At that time, and you see there was actually someone standing behind the printer and feeding it with papers, and that was the only way actually to make it work. You will see here actually that after we did the bioprinting, the cells actually, they were live and beating. So that was among the first trial that we did. So what bioprinting does for us, allow us to basically put, again, the cells that we need in the right location and have them talk to each other. So here I'm showing some of our work when we try to put different cells in different locations and see if they can actually talk to each other. So we did an implantation of that structure, and you can see whenever we put that structure in an animal, there is vascularization there, which means the cells are talking to each other and they are alive and they're going to be functional eventually. So we try to take this to a different application, printing cartilage tissue, and we actually got some functional results with that construct. This is actually part of the work that I started with Wake Forest, with the Armed Forces Institute of Regenerative Medicine, trying actually to print human skin on wounded warriors after actually extensive burn injuries. So we designed a printer that goes on top of the patient, scan the wound, and actually knows what are the missing layers and cells, and automatically deposit the cells and materials there. Within 20 minutes, your patient is ready. You bandage the patients, and you're good to go. Can you play the video here, please? So here is the scanner actually, comes, you'll see it here actually. Scan the wound, determine what's missing. It doesn't matter the topography of the wound, and then the deposition will happen right away. And as you can see, those are the cells after being laid down. This is actually, I apologize for the picture, but this is an extensive wound injury on an animal. And what we are showing here actually that we can print cells and make skin within 2 months for a patient. So application of the 3D bioprinting goes actually beyond making tissues and organs. Now, a lot of research actually using this for organ bioprinting or drug discovery or drug testing, and many other applications that we actually needs a model of the human tissue or organ in the lab, so we can do a lot of testing before we actually move to the treatment. So with organ bioprinting, it's basically the same concept. If you can take cells from the patient, this is actually the best source. If you do not, then we move to the stem cells and other cell sources. But basically, we take cells from the patient, we combine them with what we call it the bioink, or the material that actually the cells they would like to be with. And then we can do this organ bioprinting. We can do conditioning for it in the body to make sure that the cells are okay, accommodating and functioning, and then we put them back into the patient. At least there are probably more than 100 bioprinting companies around the world working on bioprinting human tissues and organs. So really the field is actually exploding when it comes to the use of bioprinting for drug discovery, organ regeneration. So as I mentioned earlier, the major breakthrough was actually last April, printing the first vascularized human heart, which is actually a right step toward basically an implantable organs. We believe we are not going to have an implantable organ within the next 5 years or 10 years. At least it's going to take us probably 20 years until we get to the point to say that we have an organ right now that we can implant in a patient. There are a lot of stuff that we need to discover and resolve before we get to that point, but we have to work very quickly. By the time I'm going to end my 20-minute talk, at least 20 people, they died waiting for an organ to be implanted, and at least 2 or 3 people, they were actually added on the organ waiting list. So it's a big problem. Everyone, I believe, in this room appreciate the need for organ transplant. And I encourage you from my position here to be an organ donor, because you can save a lot of lives with your organ donation. So as I mentioned earlier, different application drug testing. We are not created in the same way. Our tissues and organs, they are different. Our diet is different. Our structure is different when it comes to the way how our tissues and organs, they respond. So in case of a treatment, the drug that works on you might not work on me. The response that you have toward a drug or a treatment is different from someone else. So that's actually where the regenerative medicine comes to play a big role here, that we can actually have a personalized medicine. So when a patient has a certain type of cancer, we can actually test which drug works on them best, what dose works on them, and so on. So we actually alleviate that suffering that they have to go through this to for who knows if it's going to work or not. So at least we have a targeted treatment for them that is actually personalized for them to give the best results. So as I mentioned earlier, we are probably 20 years away from printing organs, and we have a lot to work on. One of the things that we actually need to figure out, what are the materials that we have to use to print those human organs and tissues. Until this moment, 99% of those printed human tissues and organs, they're actually using animal-derived materials and animal-derived substrates to grow the cells. We know this is actually not going to fly with the FDA. It's going to cause a lot of delay to ensure the safety of these materials, plus to ensure actually the compatibility between our cells as humans and the materials that we obtain from rats and pigs. So there is a lot of work actually that needs to be done on that front end. We came as a Humabiologics to basically address this gap in the market by actually providing human-derived biomaterials to expedite the development of these bioprinted tissues and organs. That heart actually that was actually printed recently was printed with a pig gelatin. And we know there is a huge difference between our materials and the human materials. So this is where we come actually to play. We are very proud to introduce the first two products last 2 months. And we are actually coming up with the first and only human gelatin that is going to be used for 3D bioprinting application, and the same for actually human collagen. We're hoping one day that we're going to be like this surgeon in the OR, that we can have organs on demand, so no one die from waiting on a waiting list for an organ. Thank you very much, and I will be more than happy to introduce, I believe, the lunch to you. So,